Dear participants, before we begin, we request the participants to make sure to unmute their microphone and off their video for the smooth conduct of the webinar. If you have any queries, you may please post it in the chat box. Good morning, one and all. Welcome to the webinar on DNA Origami, How Does Our Genome Fold, organized by Department of Zoology and Research Cell at St. Joseph's College, Autonomous, Irinjalakuda, Kerala. Our guest of honor and resource person of the day is Dr. Supriya Vartak, postdoctoral fellow, Dr. Raphael Casellas Laboratory, NIH. Before we move on to the session, let us seek the blessings of Almighty God with a silent prayer. Myself, Dr. Vidyaji from Department of Zoology at St. Joseph's College, and I'm indeed honored to welcome each one of you present here to this webinar. Now, the person giving today's session is someone I know and admire. To give a brief introduction about her, she is currently a postdoctoral fellow at Dr. Raphael Casellas Laboratory, NIH. She was an integrated PhD student at Indian Institute of Science, Bangalore. In fact, she is one of the 10 students who got selected for the integrated MS PhD program at IIC. Her research interests include DNA repair, cancer therapy, DNA structure, and currently she is working on genome architecture and maintenance. She is the recipient of many awards. Outstanding Poster Award at the 2020 NIAMS Annual Scientific Retreat, NIH USA. Best Oral Presentation Awards for different conferences held at different places. She got Best Thesis Award as well at the International Conference on Radiation Research 2018 held at Hyderabad, India. She is also recipient of prestigious Ranbaxy Science Scholar Award 2016. It's an award given to young scientists in the field of pharmaceutical sciences, and many others. In short of time, I'm cutting short the awards. Apart from this, she has more than 10 publications in peer-reviewed journals. So clearly, this shows her academic excellence, and I'm lucky to have had the honor of working with her. It is indeed a special privilege and pleasure to welcome you, Supriya, on behalf of St. Joseph's College, Irinjalakuda, and Department of Zoology, Research Cell, and everyone present here. Next, I welcome our beloved principal, Dr. Sister Asha Therese, in her absence. I also welcome all our dear faculty members, research scholars, and other participants who have joined us today. Let us now listen to Dr. Supriya Vartak on DNA Origami. Over to you, Dr. Supriya.
Am I audible?
Yes, you are.
All right, excellent. So thank you so much, Dr. Vidya, for that extremely generous introduction. And I'm truly honored that I'm giving this talk in the college. And I feel it's a wonderful initiative by the college that they invite different people to talk to BSc and MSc students, because it's always good to have exposure from what's happening outside so that you can make your career decisions or in general the path forward better. So, all right, without further ado, I'll just share my slides. All right. Just let me know if you can see the slides in the full presentation view. Vidya, can you just please let me know if the slide is seen in
Yes,
the
yes,
full presentation view?
yes.
All
Yes,
right.
yes.
All right. All right. So, thank you so much once again, Dr. Vidya and Dr. Paulos, for inviting me here. And a warm welcome to all the students to probably what's going to be your last scientific lecture of 2020. And same goes for me. So, here goes. So, today I'm going to talk to you about the whys and hows of genome folding and genome architecture in a talk which I've titled DNA Origami: How Does Our Genome Fold? Now, there were mainly two reasons why I chose this particular topic. One being, we do learn about genome folding and a little bit about the genome organization at the bachelor's and the master's level. But I feel that having studied a little bit of it during my PhD and now during my postdoc, I feel that there is this massive divide in what we learn in the textbook and to what the field has furthered over maybe the past decade. So I thought it will be a nice exercise, and it will be fun to kind of give you guys an update on where the genome folding field stands and why I think it is super exciting for me to work on. And the second reason is that, as Vidya mentioned, I am a postdoc at NIH right now, and I essentially work in the field of genome architecture and genome folding. So it was a bit of less homework for me to prepare for this talk as such. All right, so without further ado, let's get into how does our genome fold. Now, before we understand how does the genome fold, let's understand why does the genome need to fold in the first place, right? So, starting with some basic numbers, the human body has around a whopping 30 trillion number of cells, all right? And if one even begins to fathom what kind of a large number it is, my favorite kind of to go example to compare it with day-to-day life numbers is the world population at this point. So as of 2020, we have 7.8 billion people living on this planet. And if you compare the 7.8 billion numbers to 30 trillion cells, you would have to have around 3,000 such independent worlds to kind of equate the number of cells you would get through in each single human being. So it's an extremely large number to say the least. And each of these cells has its own genetic material, which is the DNA. Now, here I show an electron micrograph of a typical human cell. With at the center, you can see the nicely marked nucleus and surrounding cytoplasm with its cytoplasmic organelles. Now, it is challenging for the nature to fit all the genetic material in this nucleus, which is around 6 to 10 microns in diameter. And each DNA, if you stretch it like a rope, it measures end to end around 2 meters of length. Now, the real challenge is to fit this 2 meters worth of DNA in the tiny nucleus of 6 micron wides. Now, again, to understand it in a non scientific way, suppose consider the nucleus to be the size of a tennis ball, all right? And your DNA, which you have to fit it inside the nucleus, is made up of woolen string. So, drawing parallels between a 2 meter long DNA and a 6 micron wide nucleus, this woolen string would span for a whopping 40 kilometers. And this 40 kilometers of DNA string, you would have to in turn fit it into this tiny tennis ball. So, one agrees that there is an incredible amount of packaging and folding required for DNA to fit into this tiny nucleus. All right. So let's learn more about why this term chromatin architecture came into picture. So is this just a fancy term that scientists use who work in the field, or is it like actually an a truly marvelous DNA architecture? Now imagine that your genome is this blank piece of paper, all right, and you have to fit it in a constricted nucleus of a much smaller size. Now, an intuitive way and one of the most easiest way is just random scrumpling of the paper, which kind of reduces the size and you can easily fit it into the tiny nucleus. But this will not work for the genome or DNA because the genome is quite dynamic molecule instead of a static molecule. Why do I say that? Because it needs to change its form and structure day to day life for different processes, such as one is transcription, which is the process by which DNA makes mRNA, and followed by translation, where mRNA makes protein. Now, when each of these gene gets transcribed or the transcribing region, the chromatin has to open up or become accessible so that large number of transcription factors can actually bind in and the RNA polymerase can start the process of RNA transcription. And this opening up of the genome takes place during the transcription of if each and every gene in the human body. On the other hand, when the gene is finished transcribing, the genome has to go back to its original form. So, this is a continuously ongoing dynamic process. The second example is that of DNA replication, which is the process by which the cell duplicates its genetic material. Now, as you all must be aware, the first step in replication is to unzip or unwind the strands of the DNA such that the two strands are separated and then duplicated into in such a way that they are further duplicated into each daughter cell so that they can have their own double-stranded genetic material. Now, the third and one of the most complicated processes is mitosis. Now, you must be aware that cells follow a distinct cell cycle phase, and the main phase is characterized as interphase, followed by the four subphases of mitosis. Now, we know that in interphase, DNA exists in a decondensed phase. However, once it starts the prophase, DNA starts condensing into what we see as the mitotic chromosomes. These mitotic chromosomes are then aligned in a single metaphasic plate, and then they are pulled apart to each of the poles by spindle fibers, followed by formation of a furrow, which gives rise to two daughter cells. And at this step and even beyond, the genetic material kind of gets decondensed, and the cycle repeats all over again. And the important point to be noted here is that once the telophase is over, the cell can get the exact architecture or exact packaging or folding that it initially had back at interface, right? So, if one would imagine if I were to unfold this piece of paper, it would be very difficult for me to fold it again on the exact same folds and exact same pattern. So, scientists believe that the genome folding or chromatin architecture follows a more elegant DNA origami type of method in which the folds are precise and one can easily unfold this swan structure over here and I can refold it on the same folds and get a beautiful architecture while still maintaining constriction of size so as to maintain it in the tiny nucleus. All right. So now let's see the basic organization of the genome as given in standard textbooks. So I hope many of you are aware of this diagram which I've taken from Molecular Biology of Cell, which talks about a basic genome organization pattern across the chromosomes. Now I'll just go through it briefly one by one. So we start with what is called as naked DNA or a double-stranded DNA, the width of which is 2 nanometers. This in turn is wound around histone proteins. You have 2.5 turns of the DNA around histones, and the structure is now called as chromatin or nucleosomes. It is referred to as beads on a string because in electron micrograph images, it literally looks like a necklace made up of beads on a string. And this structure is quite stable because the negatively charged DNA is kind of balanced by positive charges of the histone. Now, where does this positive charge come from? Let's not forget histones are basically proteins made up of amino acids, and they have a lot of positively amino acids like lysine, arginine on the surface, and thus balancing the charge of the negative DNA. So this 11 nanometer fiber then folds upon itself to form 30 nanometers and 300 nanometer fiber, which kind of now start resembling a loop kind of structure. These loops then fold upon themselves to form a 700 nanometer fiber and eventually folds into a 1400 nanometer constriction, which we now know as the mitotic condensed chromosome, as I mentioned in my previous slide. So, one remarkable thing to note here is that the extent of compaction is around 10,000 times when compared to the naked DNA as shown here and between the chromosomes seen in a typical mitotic phase. All right. Now, the past decade has been witness to an enormous surge in findings in genome organization, genome folding, and genome architecture field. So, I present to you kind of a new view of how the genome is organized at multiple scales. So, to begin with, the fundamental nature remains the same. You begin with a nucleosomal scale of folding, where you have the naked DNA bound by histones forming chromatin structures or nucleosomes. We then go further into a supranucleosomal scale, wherein these chromatin structures start forming distinct chromatin loops. All right. And these chromatin loops in turn form what we call as topologically associating domains or TADs. And these TADs form together distinct chromosomal compartments, which can be transcriptionally active or accessible, which is known as A compartment, or they can be silent and more condensed, which is referred to as the B compartment. Now, further than this, you also have the nuclear scale of organization, where we find distinct chromosome territories. Now, very elegant experiments have shown that chromosomes don't randomly hang around in the nucleus, but they actually prefer certain spaces more than certain other spaces in the nucleus. And they also prefer to hang around with certain other chromosomes than distinct chromosomes. And all these forms what is known as chromosome territories. And it has been shown that these have distinct physiological relevance. And I think the past, say, the whole decade has witnessed a real surge in chromosome territory papers. Now, what I would like to focus in today's talk is explain what is chromatin loops, explain a little bit more about what is TADs, and how these TADs form chromosomal compartments. All right. Um, so let's talk about DNA loops. Now, we know that DNA is folded into looping structures, which I've shown here. So you can see loops of all different shapes and sizes bound by something here, which I'll come to in a minute. Now, how were these loops discovered in mammalian genome or human genome to be specific? So the idea dates back to early experiments in Drosophila, where people observed these huge brush-like lamb brush chromosomes in the meiotic chromosome. And as you can see here, they form distinct loops on both sides of the axis in Drosophila. And the important thing to be noted here is that these loops are quite large and they can be visualized under a bright field microscope. Now, as opposed to that, the loops occurring in mammalian genome or specifically human genome are not that big. So it's a challenge to kind of visualize their existence or even prove that these kind of loops exist in the mammalian genome. But again, there are a lot of assays or techniques which have been developed over the past decade, which have unambiguously proven the existence of such loops. And in fact, they continue to delve more into how these loops are formed and what is the physiological relevance. And just to name a few, you must have heard about chromosome conformation capture or 3C, and its updated versions 4C, 5C, high C is the latest one. So all these are excellent techniques to study chromatin architecture. Because of dearth of time, I did not include slides for these techniques, but I assure you that these techniques warrant a lecture of their own because these form the basis of studying chromatin architecture. So maybe another lecture. All right, so the million dollar question still remains, how are these loops formed? All right, so if I give you a piece of thread or a piece of rope and I ask you to form this kind of structure as seen on the left side, so that would be easy, right? But one would agree that you need some external force. For example, with the thread or with the rope, you would actually join them using your hands, using your force, and then glue it together, maybe using tape or glue. So, similarly, in case of a cell, people had an idea that there needs to be an external force which brought these loops together and which held them in place and held them in a dynamic fashion so that they could be formed and deformed as required. So, there have been many models and theories on how these loops originally formed, and I think the most widely accepted one is that of DNA loop extrusion, and it has huge experimental backup and is the most widely accepted in the architecture field. So, what exactly is extrusion, and what do you mean by the DNA loop is extruded? So, the literal meaning of the word extrusion is shaping or forming something by forcing or pushing through a small space. All right. So it's like threading a needle through the small head of the needle. So, what I'll do is I'll walk you through the steps of DNA loop extrusion. Now, it's a fairly complicated process, but I've made it simpler for you guys to understand it better. Now, imagine that this is a strand of the DNA. All right, the blue line is the DNA. You have three key players who are responsible for DNA loop extrusion. One is the cohesion rings, which is actually the extruding complex. The second is the green guy over here, NIPBL, who actually loads cohesion rings onto the DNA. And the black pillars seen here are CTCF. They are the stop signs, which tell the extrusion to stop at that particular position. Now, as I told before, NIPBL signals the cohesion to load onto the DNA. And you can again imagine this as threading a needle and pulling the thread from the needle. You have the DNA coming out, pulled from here now, which is extruded and extruded to such an extent that it forms a full fledged loop domain. And these anchor points, pillars which were far away, now come closer because of the loop extruded in between. All right. So, just important points to be noted that this loop formed is called as the loop domain, and the base at which the rings are still intact and the CTCF signal stands is the loop anchor. Now, this was a very rough explanation of what extrusion is. I'll come to what each of these factors or each of these players does. Right. Now, coming first to talk about NIPBL. Now, this has been considered as the loader of these cohesion rings, which actually form extrusion. NIPBL, which is called as nipped B cell protein, is a protein which loads on, which recruits the cohesion and stabilizes the extruding structure. Now, it was quite an exciting year for NIPBL. I can definitely not say otherwise. But just for NIPBL field, we had the cryo-EM structure of human cohesion NIPBL DNA solved, and it looks something like this. So the cyan or light blue colored ribbons that you see here is what NIPBL looks like, and behind it is cohesion, and going through in the middle is the double-stranded DNA. So you can see the position at which it binds DNA. Now, visualizing NIPBL structure with cohesion on DNA and understanding its mechanism becomes important not only for basic sciences, but it also becomes important for human health and disease. NIPBL mutations have been previously reported to cause a syndrome known as Cornelia de Lange syndrome or CDLS. And I'm so sorry to show these pictures, but this CDLS is a developmental disorder present right from birth, but it is not inherited. The occurrence is estimated to be 1 in 10,000 births, and the children who have CDLS, it affects their overall growth, development, intellectual, and learning abilities. Now, the interesting point is that around 60% of these patients were found to harbor specific mutations in the loader protein NIPBL. All right. And if you look at the spectrum of the disease mapped from mild, moderate to severe, you can see that NIPBL missense mutations and NIPBL truncations, which are just different types of mutations occurring in the protein, they span the severe to moderate phenotype of the disease. And the rest of the chunk comes from, of course, in the mild, you have the NIPBL mosaicism as well, again, another type of mutation, and also mutations in SMC1, SMC3, RAT21, etc. Now, one would be curious to know what these other proteins are and why do they give the same CDLS phenotype as that of the loader or NIPBL protein, right? And nature does not cease to amaze us because these proteins that I mentioned before actually form part of the cohesion complex or the extruder machinery. All right. Now, although we show them as these nice ring like structures, the actual structure is something like this, where you have different proteins coming together to form a ring like structure around the DNA. You don't have to remember or go into details of what the names are of the each, but just keep in mind that it's formed by these two green structures, which are ATPases, all right, SMC1 and SMC3. So they are like motor proteins which use the energy of ATP to actually physically extrude out the DNA from between the ring like structure. You can also see the loader, NIPBL, and the accessory protein MAU2 in this case. And through the electron micrograph image, I think we have a fairly good idea of how this protein looks inside the cell and how it functions inside the cell, being an extruder molecule. Now, to understand this better, again, my favorite to go example is the sugarcane juice extractor. And I'm sure all of you are very familiar with this. It is one of my personal favorites. I imagine that the person who puts the sugarcane in is the NIPBL who loads the sugarcane onto the machine. The sugarcane getting actually extruded out is the DNA, and this machine, equipped with its own set of wheels and motors, not using ATP but electricity in this case, is the cohesion molecule. This process is much more complicated, but I find this analogy the closest to a working cohesion model. So now that I've told you this, one can go back to the time when people did not know that cohesion was actually responsible for extrusion, and one can ask, how did people come to know that cohesion is actually responsible for extrusion, or that cohesion is actually present near extruded chromosome or near loop formation? So, a little history about cohesion, and right from starting from its name cohesion, where did the name cohesion come from? So, it all stems up from sister chromatid cohesion, a phenomenon which is very common inside every cell, happening during every cell cycle. Now, what happens is, as I mentioned before, each cell goes through G1, which is for gap, G1S, followed by S synthesis, followed by G2M. So, in synthesis, as I mentioned before, it gives rise to duplication of the genome, and the two sister chromatids are actually held together by cohesion. The literal meaning of cohesion is sticking together or holding together. So, this is what the original function of cohesion was, that to hold together two sister chromatids produced in S phase. Now, why was it important for the sister chromatids to be always held together, right? Now, this importance was realized only in not in S phase where it actually happens, but only in the later mitosis phase, where if they are not held together, it leads to a big problem. So I already mentioned this process before. Uh, Prophase chromosomes are formed, they are aligned at the metaphase, they are kind of pulled apart equally by the spindle fibers, and the two daughter cells get equal amount of genetic material. Now, early experiments in yeast showed that there were a set of proteins which now we call as cohesin that were responsible for a process called as bi-orientation. That means arranging these at the metaphase plate in such a manner that they would be equally pulled apart so that both the daughters get equal genetic material. Now, these experiments also include mutant yeast strains, which had no cohesin. All right. So, what they observed was if there was no cohesin, the sister chromatids were not held together, and therefore they were not placed in a organized manner at the metaphase plate, and there was unequal distribution between the daughter chromosomes. And you can imagine the downstream disturbances caused in each daughter, because getting an extra chromosome or getting a less chromosome would be detrimental. Now, although this was a big breakthrough for cohesion's function to hold sister chromatids together, what next? So, how did people come to know that it has additional roles in architecture or loop formation? Now, the biggest clue came from studies in neurons. Now, neurons, as you all must be aware, are terminally differentiated. All right. So they don't undergo cell cycle like other cells, and they do not undergo mitosis once they come into being. So technically, they would not need cohesion at the sister chromatid step because they don't need it for mitosis at all. But studies in neurons showed that these cells had very much active cohesin in large amount, and cohesin did bind to DNA. So the question was, what is cohesin doing in these cells if it is not aiding in sister chromatid cohesion? And that's where an additional role of cohesin stemmed up, and um, people hypothesize that if cohesin can bring together two pieces of DNA in trans, it might also be able to do this with two pieces of chromosome which are in cis, and hence aid loop formation. All right, now, so we have the NIPBL loading onto the DNA. We have the cohesin rings loaded. You have the extrusion happening. Now, how does cohesin or the cell know where to stop and how much DNA to extrude? All right. Although I've chosen to show only this much amount of DNA in the diagram, what signals these cohesin to actually stop at this region, and why doesn't it just keep extending over DNA distance? Now, early clues of this came in when scientists zoomed in on this area and checked what exactly is happening at this area. And to their surprise, what they found was in this region, you almost always had a protein called as CTCF bound at the very ends of the loop anchor. CTCF is a transcription factor. It stands for triple CTC binding factor, and it can bind specific sequences inside the genome, which are called signature sequences or DNA motifs, which I show here. Now, one can recognize that if this motif or signature sequence occurs on one strand of the DNA, if it runs from the 5' to 3' direction, the CTCF binding can be considered as in a forward direction. And if this sequence appears on another strand running from 3' to 5' direction, CTCF can be reverse facing. So consider a stop sign which you are seeing from the same direction. So you are facing forward, and the same stop sign I'm viewing from the reverse direction, and I'm facing reverse. Now, there are four possibilities of how two CTCF motifs can be found on an entire linear DNA. And this is just common sense, where the four possibilities can be either AA, that is, both are facing forward, it can be AB, one is facing forward, one reverse, it can be BA, or it can be BB. All right. Now, interestingly, what was found in experiments was that the extrusion stopped only if the signs were in this scenario, which is one was forward facing and one was reverse facing. And in all the other cases, 1, 3, and 4, the extrusion just continued until it came up to a forward and reverse facing CTCF signs, and then it stopped. So this is known as the CTCF convergence rule, which essentially stands for convergent or opposite side facing CTCF motifs. All right, now that we're equipped with the basic key roles, let's look at a small video made by Rice University Aiden Lab, which shows the central two rings here of cohesion, which is extruding the loop away. Now, this extrusion will happen until it encounters a CTCF motif as shown here. So, there goes the forward motif. Now, it will stop only if it encounters a motif in reverse. So, this is in the same direction, hence it passed. This is in the opposite direction, and thus the extrusion stopped here. And then you can see that this becomes the base of the loop, which is called the loop anchor, and the loop on the right side becomes the loop domain. All right. Now, climbing one step higher, the organizational ladder, let's see what this whole loop domain, how does it exist inside a cell, and what is it called? So we now come to what is called the topologically associated domains, or TADs, which essentially is formed by multiple loop structures which are in close proximity in 3 dimension but not in 1 dimension. All right, so they can be KBs away on a linear scale, but because of extensive folding and loop formation, they come in proximity in 3 dimension in a cellular nuclear volume. Now you can have active TADs as shown here, marked in green, which are more open and transcriptionally active, or you can have repressed TAD segments, which are essentially groups of loops which are not transcriptionally active and they are little condensed. So zooming in on this particular TAD, basically a TAD serves as two functions. One is that it increases the chances of interaction of promoter enhancer within itself, as seen by this green triangle. And second, it kind of insulates or prevents the interaction of this enhancer or promoter from the neighboring TAD or the neighboring green oval shown here. So I like to see it as any fourth wall that is built, where you would find maximal interaction of people within it, and interaction from of people within is actually prevented from interaction of people outside. So it serves as the walls of a fort. You can compare it that to like that. Now, this might get a bit confusing because I just dived into what is called as transcriptional regulation here, right? I did mention that these enhances the interactions and this prevents interactions. So, what are these interactions that I'm talking about? Now, to understand this better, I again go back to the basics, where the central dogma of molecular biology tells us that DNA forms mRNA because of transcription and mRNA forms protein via translation. Now, the process of DNA and DNA forming mRNA is extensively regulated. And I think understanding this with respect to architecture becomes quite important, which will be clear in subsequent examples. Now, how is this regulation brought about? The main elements here are a promoter and an enhancer. So, a promoter, you all must be familiar with, is a region where transcription starts. And although in bacteria, the RNA polymerase can directly bind at the promoter and start transcription, in higher organisms, this has to be aided by various accessory factors like transcription factors, mediator complex, etc. Now, what are enhancer elements? So, these are specific genetic elements which are present maybe kbs apart from the promoter. They might be upstream or downstream, and they literally act as this regulator of the fan, as shown here, and they control the speed or they control the efficiency at which genes are transcribed happening at the promoter. Now, how does this happen? How does a KB's away enhancer actually control what is happening at the promoter? And I've chosen a series of three examples which will make clear how extrusion kind of plays a role in architecture, and because of extrusion, how different enhancers comes in close proximity to the promoter, thereby regulating this fan switch. So I call it the spatio-temporal regulation of genes. Now, these three examples, as you'll see, are rated in their higher order of organization from lower to higher. So, in the first example, enhancer promoter interactions and the way DNA architecture shapes the genome is important for cell fate decision. All right. So, to understand this concept clearly without having specific names of genes to cause more confusion, I've just named it gene X here. Now, gene X is responsible for, is present in both the cells. This is a neuron and this is an epithelial cell. However, when gene X comes in close proximity to an enhancer A, all right, and this is happening via differential extrusion. So, again, think of it as a thread going in a needle, and the needle is actually controlling how much thread has to be pulled in order to bring gene X close to enhancer A. Now, it was observed that when gene X comes in close contact with enhancer A, it drives the selection of the cell in such a way that the cell is differentiated into a neuron. And you can see that the blurred structure here is enhancer B, which is not really uh, important here as it is not coming in close proximity to gene X and it is not regulating gene X promoter. On the other hand, now in this scenario, you see that the extrusion has happened to a longer extent, and actually enhancer B has come in close contact with the promoter gene X. And the result of this is that the cell has shaped into a epithelial cell. And again, in this case, enhancer A.
Excuse me, Dr. Supriya.
Yeah,
Your
yeah.
presentation is gone. Could you just again make it into
Oh,
presentation mode?
sorry. All right.
Someone else had presented. That's why.
Oh, no worries.
Sorry.
Was
Yeah.
it now or from a little bit
Just for
before?
30 seconds.
Oh, okay. Okay. Can you let me know if it is visible, please?
Yes. Now it's fine. Yeah.
All right.
Thank you.
Thank you. Thank you so much. All right. So, just a brush up maybe of the last 30 seconds where we saw that enhancer A coming close to gene X was important for a neuron development, and enhancer B coming close proximity with gene X was important for epithelial cell development. All right. Another example, now this was with respect to cell fate decision. Another example is in Drosophila embryo, and you can see a nice Drosophila embryo here with anterior and posterior side. And these are the segmentations for required for development. Now here, a central gene, Eve, named here, is controlled by 5 different enhancers as shown as yellow bubbles in this case. And you can see that the cohesion ring shown in brown here specifically brings, suppose, the enhancer 3 or 7, 3/7 near the promoter Eve, you have activity in stripes 3 and 7. On the other hand, if the cohesion extrudes the loop further, and now the enhancer 4/6 is in proximity with the promoter, you have activity in stripes 4 and 6. So, this way, the whole segmentation of the embryo is controlled, and you have expression of different genes from the same promoter but different enhancers, owing to differential extrusion by this cohesion molecule. And an example three would be indirect in work in humans, where people have shown that in human embryo development, you have several enhancers, again focusing on a single promoter in a developmental gene. Now, I've placed a X here because if there is a mutation or any abnormality in this developmental gene or promoter, you will have embryonic lethality, because this is absolutely essential for survival of the human embryo. If, however, in any of the enhancers have mutation or some abnormalities, for example, in this case, 1 to the third enhancer has a mutation such that it cannot regulate the expression of developmental gene, since this is specifically required for normal development of the face region. Patients who have abnormalities in or mutations in this enhancer often face orofacial clefts. So overall, this gives a nice idea of how chromatin architecture comes in close contact with promoter enhancer interaction, and how this is eventually important for physiologically relevance and of a distinct phenotype. All right, now coming to the last part of the talk, we are almost to the end of the organizational ladder. Now we come to A or B compartments. All right, they sound quite simple, but I think that the most unstudied and unexplored regions within the nucleus right now. So essentially, these are comprised of entire chromosomes existing in particular compartments in the nucleus, and they are just simply named as compartment A and compartment B. You also must be familiar with another names of compartment A called as euchromatin and compartment B called as heterochromatin. Now, it is observed that generally chromatin A is quite accessible or is quite open and constitutes a large part of the nucleus. And it usually is transcriptionally active. So there is a lot of expression of genes going on from this segment of the DNA. On the other hand, compartment B is kind of condensed and it is not very transcriptionally active. Another interesting point came from visualizing these heterochromatin and euchromatin in the nucleus. And you can see from this picture that the euchromatin, which is lightly stained, is more or less in the center of the nucleus, or one can say it occupies the whole section of the nucleus. And the darkly stained region is the heterochromatin, which, apart from a few exceptions in the middle, most of it kind of nicely lines up the nuclear envelope and is mostly situated at the periphery. Now, again, there are a lot of exciting papers on why this is the case. Why is heterochromatin at the periphery? Why is euchromatin at the center? And is there ever an exchange of genetic material or transcriptional activity between heterochromatin and euchromatin? All right, it's quite fascinating. So I think with this, I haven't checked the time, but I hope I'm within the time frame. And I'll just give a quick summary. So we learned that human genome is not randomly scrumpled like a piece of paper, but is neatly organized at multiple scales, like this swan origami that I show here. DNA folds into loops and is formed by a complicated process of extrusion. And don't forget the sugarcane juice example. DNA extrusion is carried out by cohesion complex, the three main players, cohesion, the loader NIPBL, and of course it is stopped by CTCF stop signs, and also remember the convergence rule in this case. Fourth, loops further forms topologically isolated associated domains or TADs, which are critical for transcriptional regulation. And multiple chromosomes exist in distinct nuclear compartments formed, which are called as euchromatin or heterochromatin. So, with that, I hope you found the lecture interesting and learned something. And a big thank you from my side. I will be happy to take any questions. And if not, then I wish all of you a very happy and scientific year 2021. And if no questions now, you can also reach me at the email ID that I've given here. And thank you all again so much.
Thank you, Dr. Supriya. It was a very interesting topic, and your presentation made it more interesting as well. So, thank you for sharing your knowledge with us. And now the session is open for discussion. If you have any queries, you can post it in the chat box or you can ask directly.
If no questions, then I'm going to start asking questions. That's my rule.
Dear students, I know you have a lot of questions in your mind, so you can just ask. It's okay, be it very complicated or simple, anything you can ask. You have the best person available now.
Any question on genome architecture, actually.
It's okay. Let's give them a break. It's the last topic of last lecture of
Oh,
the
there's one
year.
question, actually. There's a question by Arun Kumar. Is there any relation between the incorrect positioning of chromosome in its own nuclear territory in some genetic disorders and incorrect DNA origami?
Yes, that's an excellent question. Um, So right now, I can't think of any examples where there is a disease phenotype which is caused because of that. I know for sure that if an enhancer A is supposed to bind to gene X, suppose is supposed to come in close contact and regulate that, and instead of that, if an enhancer B now regulates the promoter, there can be different phenotypes. So these kind of studies have extensively been carried out in Drosophila, and you have all sorts of mutations and abnormalities happening. But if you ask me like a disease relevance such as CDLS or the Cornelia-de Lange syndrome, that I cannot think of right now, but I can get back to you on that one. But to answer your question, yes. I mean, if a wrong enhancer falls at a wrong promoter, you definitely have a lot of phenotypic abnormalities happening. Great. Thank you.
Any other queries? Okay, so Priya, then I have a question. So
Yeah.
does this proteins like NIPBL protein, do they play any role in other non-BDNA structures? Like, do they have any role to play in formation of other non-BDNA loops or structures?
Yeah, that's a good point, and I think it's very dear to us because we've studied long DNA structures. But to answer your question, there have not been studies. So, as I told you, this field is quite, I mean,
New
in
object.
infant stage, yeah. So you can imagine, right? In the cryo EM structure of NIPBL, human NIPBL, came out just in June 2020.
Yeah.
So we have just begun to visualize how the structure looks, and now I think there'll be quite a bit surge of how this NIPBL is involved in non-BDNA structures.
Okay. So once this loop is formed, how is it resolved later? Like once it has to come out, right, from this loop structure later.
Right, right.
So this proteins are just coming out of the DNA and then the loop is resolved, or is there any other?
Yeah, again, so those are, um, I mean, as of now, people think it is by the action of helicases,
Mhm.
where So once the ring structure has bound, and it's like a curtain, right? If you thread a needle or a curtain, the DNA which is downstream will get entangled.
Mm-hmm.
And also at some point you need to bring back the loops and dissolve the loop. So that is by the virtue of helicases, which actually will cut the loop
Okay.
slightly around and then bring it back to its original form.
Okay. Okay. Thank you, Supriya.
Thank you. All right. Thank you so much.
Okay, there's one more question
Yeah.
by Anukumar again. DNA origami has been used for construction of molecular nano robots, isn't it? What do you think about the future applications of nanobots in medicine and drug delivery?
Oh,
Nanorobots.
wow. Yeah, interesting. So, yeah, to be honest, I don't know the answer to your question because I know that there are a lot of artificial DNA structures based on the origami and folding. But with respect to what future does it have for nanorobots, we just have to wait and find out, I think. I mean, of course, I can read more about it and get back to you on this one, but yeah, that's interesting. Great. Thank you.
Okay, so thank you, Dr. Supriya, for clearing our doubts. Now, as we are concluding the session, I invite Dr. Gigi Paulos, head of the Department of Zoology, to propose a vote of thanks. Over to you, ma'am.
Yeah, thank you, Vidya. Very happy to propose the vote of thanks of this webinar. Let me begin by thanking Almighty for bringing us together and giving us a chance to interact with Dr. Supriya. Um, Dr. Supriya has given a wonderful briefing. It was a wonderful webinar. I'll tell you, the session was very great in terms. It was in depth and it was in simple way that you presented it. And I'm sure the most of the students and the faculty who were attending had now they have got a better idea of how things work at the DNA level and this origami, and it's really interesting. And we wish you that your work progress well and you come out to us again. Okay, so thank you so much for being with us, and it's late night there, still you took out your time from your busy schedule and you gave us this talk. So thank you so much on behalf of St. Joseph's College, on behalf of the Department of Zoology and the Research Center here. Thank you, ma'am. Next, I would like to
Thank
thank
you
our
so
dear
much.
principal, Dr. Sister Asha Teres. She couldn't be here with us as something urgent came up. Otherwise, she would have joined. She would definitely wanted to meet you in person. I mean, in the virtual way, but she couldn't. So she has given her regards to you. And I thank our dear principal for giving us an opportunity to have this webinar. I also take this opportunity to thank Dr. Sister Flaret. She is the director of the research center here. Again, she is not here, but in her absence, I thank her. I thank all the dear participants, distinguished faculty members, the research scholars, many of the students who have joined us in today's webinar. Thank you for joining with us. I also want to thank the team behind this webinar. Dr. Vidya is the coordinator of today's webinar. I thank Dr. Vidya. Then we have from the department, Ms. Deepthi, Ms. Shibita, Ms. Juliet, everyone associated with us. Ms. Sneha is also there, working behind the scene. And thank you all. And once again, thanking one and all, I conclude and I also wish you a very happy and scientific new year. Thank you.
Thank you,
Thank you
ma'am.
so much. Bye bye. See you.
Thank you, Supriya. So
Thank
we
you,
have
doctor.
posted we have posted the link of the feedback form in the chat box. Kindly submit the form to receive your e-certificate of participation within a week. With this, we have come to the end of today's webinar. Thank you for joining with us. Wishing you all a very happy new year. Stay safe. God bless. Thank you, Dr. Supriya.
Thank you. Bye.